Hello, my name is Audrey Grotheer, and today I will be talking about the effects of the insertion of a Huntington polyQ transgene on the geotaxis response of Drosophila. It is important when discussing research to keep in mind why it was performed to begin with. This experiment was designed to observe the geotaxis response in fruit flies, their instinct to climb up to the top of any container they are in. But in that observation, we hope to find information that can be applied to the study of Huntington's disease in humans. Huntington's disease is a neurodegenerative disease that is 100% lethal whenever it is present. If an individual expresses more than 40 glutamine repeats, we can say with certainty that they will develop the disease. Our work with Huntington's and even our work implanting the HTT gene mutations in fruit flies is part of working towards a goal of better understanding the progression of the disease and ultimately finding a treatment for it. In this specific experiment, we were building off work we did previously in the semester of using an actin driver to express 16 and 128 CIG base pair repeats in flies. From that research, we found that expressing only 16 glutamine repeats had little to no effect on fly behavior, but expressing 128 glutamine repeats was in most cases lethal. In this experiment, we used an LV driver to express the mutation only in neural tissue, rather than in every cell of the body. To do this, we crossed LV flies, male flies, with virgin female UAS Q16 and UAS Q128 flies. In the GAL4 UAS system, as seen in Figure 1, a protein produced by the GAL4 gene, which is also called GAL4, must bind to the UAS regulatory sequence in order for the HTT transgene to be expressed. In this way, we were able to spatially and temporally specify when expression took place because a fly had to be of a certain genotype to express the HTT polyQ gene. After 3 weeks, we conducted our experiment to observe how long it took for the first fly in a vial to climb past a line 1 inch from the top, and how many flies after the first could cross within 10 seconds. You can see here in Figure 2 how the experiment was set up. In each vial, we had approximately 10 flies of the same genotype. 1 vial at a time, we knocked the flies all down to the bottom and started a timer when they began to climb back up. We recorded the time at which the first fly crossed this red line and then counted the number of flies that crossed within the next 10 seconds. We found that flies expressing Q128 repeats were affected most by Huntington's disease. That is, they were the slowest group for 1 fly to cross the line, and they were the group with fewest flies total to cross within 10 seconds of the first. This was not surprising and fit well with the results from our previous experiment regarding HTT transgene inserts in Drosophila. In that experiment, we found that Q128 flies died within a few weeks when they expressed the mutation in every cell of their bodies. Here, we found that severe motor impairment due to expression of the mutation only in their nerve tissue. This impairment took the form of slower individual flies and fewer flies out of the group who were able to climb at all. The average time for the first fly to cross the line was 7.84 seconds, which, as you can see, was much higher than the other averages. The average percent of flies to cross within 10 seconds of the first was 28.8%, which, again, as you can see, was much lower than the comparison groups. From these results, we can clearly see that Huntington's disease affects the Q128 flies the most. They were the slowest to climb the vial, and the fewest total climbed at all. Comparing the averages attained for the Q128 expressing group with the Q16 expressing group confirms that additional CIG repeats in the HTT gene correspond with more severely impaired behavior in fruit flies. The Q16 expressing flies had little to no change compared with the Q16 non-expressing flies. In fact, the average time for the Q16 expressing fly group to cross the line was faster than the average of the Q16 non-expressing flies. Although we did not have sufficient biological replicates for these categories, these data indicate that there is no significant effect of 16 glutamine repeats in flies. To sum up, by examining the geotaxis response in fruit flies expressing glutamine repeats in neurons, we were able to observe some of the physical effects of Huntington's disease in a model organism. We observed confirmation that more glutamine repeats are linked with more severe motor impairment, which has been found in humans as well. Hopefully, future experiments will continue this research in other model organisms to find results even more applicable to the human disease. Thank you.